biologist, Miss Ray here. Today we're going to be talking about protein synthesis. Now, in lesson 1, you learned about the structure of DNA. In lesson 2, we took that structure of DNA and then we replicated it into more strands of DNA. And now we're going to take that DNA and we're actually going to turn it into something on actually how an organism uses that information, which is it needs to make protein. So we're going to take the DNA, turn it into RNA, and then translate it into protein. It's complicated. It's going to take some practice. We're going to be at it for several days, but I know you can do it. Remember, if you have any questions, ask help. Let's do this. Yay! All right, let's get started with our Unit 4, Lesson 3 notes on protein synthesis. Please have your packets ready and let's get started. So first, we're going to talk about building proteins from DNA. This is the overall arching concept of this whole entire unit. We call this central dogma. And the whole entire point is that why do we have DNA? DNA is our blueprint, tells us sort of what our cells need to do, but it needs to be able to pass along that information. And to do that, it makes proteins that, again, help our cells do its necessary functions. So central dogma pretty much goes DNA, we make DNA, we replicate it, we then transcribe it into RNA, and then we translate that RNA into protein. And our end goal is to, again, take the DNA, turn it into protein to, again, help the organism do what it needs to do. So we're going to first start talking about RNA. We talked about DNA in our previous lessons, and now we have to talk about RNA, and mainly the differences between RNA and DNA. So first off, let's define RNA. RNA is ribonucleic acid. It's the R and the N and the A, and that's how it gets its abbreviation. And this is just another type of nucleic acid with a variety of important jobs. There's actually multiple types, and we're going to get into those. As you can see, there's even already an obvious structural difference in RNA from DNA. So let's talk about the differences. First off, RNA has the sugar ribose. Remember, DNA has deoxyribose. So this one has ribose sugar. It also is single-stranded. Remember, DNA is a double-stranded helix. Even in this image before, you can also see it's just one strand. It has those nucleotides, it has those nitrogenous bases sticking out the side, but again, it is just one strand. And lastly, DNA has the A's, T's, G's, and C's. RNA does not have thymine. There is no T. Instead, we replace it with U. And that's one of the biggest ways we can tell, is that a DNA strand or is that an RNA strand? If it has a U, it's got to be RNA. So let's talk about the types of RNA. There are 3 types. Okay, we have messenger RNA, which is the one that we'll be dealing with the most. And this is the one that carries the copies of DNA's instructions to the cell. It's like a blueprint that took it from the architect to tell the builders how to actually build it. So again, it's the blueprint. As you can see, you have that single-stranded sort of thing that we saw previously on the other slide. Now you have ribosomal RNA, also known as rRNA. This one pretty much is the ribosome. If you see the ribosome, that is actually rRNA. And remember that a ribosome's thing is to make proteins. And again, that's a whole entire point, is it takes the template that's passed along to it and it makes proteins with it. Lastly, we have tRNA or transfer RNA. And this is the thing that takes, again, the information using mRNA and rRNA and then delivers amino acids to build, again, strands of amino acids that we call polypeptides, which are proteins. It gets its name, if you look at that shape below, it looks sort of like a T. And so, again, that's where we get that sort of T shape. And again, it's transfer RNA because it's transferring and moving things. Now, transcription. So remember, we've done DNA replication. So we've taken a strand of DNA, we've replicated it, and formed, again, a new strand of DNA that's part old, part new. Now we have transcription, where we take a DNA strand and we transcribe it. Think scribe. Scribe means to write. So we are taking that DNA strand and we are writing something else. It's similar. Maybe not exact. Maybe it's abbreviated. Maybe it's a shorthand. It's pretty much the same thing, but it's also writing. So this is the process in which segments of DNA or genes serve as templates to produce complementary RNA molecules. Now, they're not exactly the same as DNA. Remember that RNA has uracil and DNA has thymine. But again, it is complementary. It pairs up with a DNA strand. Now, where does this occur? This occurs in the cell's nucleus. It has to occur there because it uses DNA, and DNA does not leave the nucleus ever. Now, there is an enzyme, just like we had an enzyme that helped, a couple enzymes that helped us with DNA replication. The main enzyme you need to know here, there's much more, but the one we want you to know is RNA polymerase, not to be confused with DNA polymerase. Now, let's look at the 3 steps. First step is just like DNA transcription, we have to unzip the DNA. Step 2, the RNA enzyme, the polymerase, is going to start bringing nucleotides and adding them to it, just like DNA polymerase did, but this time it's with RNA nucleotides. And it's using the DNA strand as the template. And here's where our new base pairing rule occurs. If the DNA has a C, you pair it with G. If the DNA has a G, you pair it with C. If the DNA has an A, instead of adding a T, you add a U. And then, of course, if it has a T in the DNA, you'll add an A as normal. Remember that the RNA strand has to be made up of only C, Gs, As, and Us. And step 3, we're going to separate the RNA strand from the DNA because the RNA strand needs to be able to leave the nucleus and the DNA strand has to stay there. Now here's, again, an image of this happening. As you can see, you have that double helix that's being unzipped. We have that enzyme, the RNA polymerase, that's, again, allowing the nucleotides to be sort of added. And as you can see, you have this single-stranded messenger RNA that is being made using the template strand of DNA. Now, you'll notice that there's only one strand of DNA that's actually being used as a template. Okay. You'll also notice that, again, specifically, basis to use messenger RNA or any kind of RNA is, again, A's, G's, C's, and U's. One last thing I want us to take note is that sometimes you're going to have to identify what's DNA and what's RNA. Again, if it's double-stranded, it's DNA. If it has thymine, it's DNA. If it's single-stranded, it's RNA. And if it has a U or uracil, it's going to be RNA. Now, we need to practice. So in your packet, you do have 2 practice problems where you're being given a strand of DNA, and now we have to just try to describe it. So let's do this. So first off, let me move my picture. There we go, just in case we need to have the base pairing rules right there. Okay, again, we have the C's and G's go together, but now we have the A's and U's. So you'll notice that I formed these in groups of 3. We call those codons. We'll talk about them later. But it makes it a little bit easier and you're less likely to make a mistake when transcribing. So the very first one that we have is you have that TAC. So T is transcribed as A's usually. Then we have that A, instead of a T, we give it a U. And the C, we give it a G. We go to the next one. A's transcribe into U's, G's transcribes into C's, C's transcribe into G's. T's, again, we're making, but remember, T's can be in the DNA strand, but we're making the RNA strand below. That's the one that's below that we're making. So it's okay to have those, again, T's turning into A's. And then we have that C that turns into a G. Then we have the CUG from the GAC, and then lastly we have AUG. Okay, I want you to pause, see if you can do the practice strand number 2 yourself. Remember that when you see a T, it will transcribe into an A because A's are in messenger RNA. But if you have an A, you're going to transcribe that into U. There's no T's that you're going to be writing here. All right, let's see how you did. All right, make sure that you copy this down and let's move on. All right, now that we have successfully transcribed, we've taken the DNA, we've transcribed it to messenger RNA, and the messenger RNA can now leave the nucleus, get to the ribosome, and actually help make proteins. We need to do that. So the messenger RNA goes in, attaches to the ribosome, which is that rRNA, and with the help of the tRNA, we build this strand of amino acids. Now, instead of transcribing, which means we're writing and using nucleotides that seems very similar, not exactly the same. This time we're going to translate it into completely something new. Remember, if you're translating something, it's usually going from one language to another language, right? And they're not similar. It's not like you're summarizing or rewriting notes like we did with the DNA to messenger RNA. This time we're taking the information and we are turning it into something new. This is called translation. All right, so translation is the process in which the messenger RNA message is decoded to make a protein. Now, this is going to occur in the ribosome. Remember that that's the protein making machines, and this will be outside of the nucleus. Now, again, look at this sort of thing, this tRNA. You have a couple things. You have that amino acid that will be attached to the very top as it's being delivered. And you'll see this anticodon that has nucleotides, again, RNA nucleotides. And it's going to match up with those messenger RNAs and pair up, and it'll drop off the amino acid and bring some more. Okay? Now, just like we have, obviously, a couple other types of RNA, let's talk about how we're going to be using those. So we've used messenger RNA with transcription, but we're now going to see tRNA being used. And we're in that tRNA, you have this anticodon. Remember how when we were transcribing in practice, I put them in groups of 3? Those groups of 3 are called codons. The tRNA has an anticodon that pairs with each of those codons of the messenger RNA. We also have the rRNA, which is the ribosome. Now, in a messenger RNA, the instructions for building a polypeptide or a protein are RNA nucleotides. We're using the A's, the U's, the C's, and G's. And they are read in groups of 3, just like I mentioned before, and we call these codons. So as you can sort of see, you have in this image below groups of 3 nucleotides. And they are then translated into methionine or threonine or glutamate or leucine or arginine or serine. And as you can see, those are very different than our nucleotides. Again, we're translating, not transcribing with this. Now, there are some steps that you need to know. These are already written in your notes, but it is important to note that although you may not have to write these out, you need to be familiar with them because you are going to have to do them. So first, we need to know that step 1, the ribosome attaches to the messenger RNA. Okay? Step 2, we're going to begin at AUG. AUG is sort of the start. I want you to think of AUG as August, and that's sort of where we sometimes start school, or at least that's when teachers come back. So again, that's the sort of start. We'll always start there. So you're going to look for that AUG on the messenger RNA. We know it's RNA because it has that U in there. Now, the tRNA anticodon binds to that AUG, and it brings the very first amino acid, and it's always going to be methionine. So that's going to be something that's really easy. The very first one, you find the AUG, the tRNA brings the methionine to it. Step 3, the tRNAs continue to deliver these amino acids and continue to add them to a chain that can be very long. And when it finally reaches, you'll see what I'm talking about when we actually get to practicing, but you're going to come to a point where when you're coding them out, it'll say stop. And guess what you do? You stop. It doesn't matter that there might be nucleotides after that, but you will stop. You will not need to go on further. All right, so couple different things. How do we translate the language of DNA into the language of polypeptide? We need a genetic code. Again, that's the language that we first start out with DNA and RNA. Those are the A's, the G's, the C's, the U's, and the T's. These nucleotides, that is a language. They're formed into these words, which we call codons. And then we use this tool called a codon wheel. And as you can see below, I gave you a lot of different codon wheels. You have specific codon wheels. You'll always be given a codon wheel. It's never something anybody has to memorize. So you'll always be given these tools. You have one in your note packet that you'll be using during class. You can see how there are a lot. Some of them go into more detail. And then at the very end, I want you to notice that, yes, you have a what we call it's not necessarily a codon wheel, but a codon table. We find that in high school, specifically 9th grade, it's easier to use a wheel rather than the table. People make less mistakes. So that's why we use the wheel. Now, let's do some practice with it, all right? All right, so first off, we already know how to transcribe. So you take the DNA sequence and you turn it into RNA using your base pairs that were mentioned over here, the C's and G's and the A's and U's. Chances are you rarely will actually have to do, unless you're specifically just doing DNA replication. Once we get to protein synthesis, you're always going to be using those base pairs. So when in doubt, always write U. Okay, now what you're going to need to do is that we're going to have to translate this into an amino acid sequence. And to do that, we need our wheel. You have your wheel in your notes. Okay, and how we do this, and you have directions that walk you through for an example, but this is what you do, is you start in the very center of the circle. So we find the first letter, A. So now we know what quarter of the circle we're going to use. We find the second letter, U. Our second letter is right here, the very bottom, we have U. And then the last letter, G, which codes specifically to MET, which means methionine. And we do that with the rest of them. A, G, C. So we start out with A, G, C, and that codes out to serine. We have U, C, G, serine. You'll notice that if you actually look around the codon wheel, there are several ones that are actually repeated, and that's okay. It's redundant, and actually that's sort of helpful. So sometimes when there's mutations, things don't actually change. So then you have G, 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 that's glycine. Then you have A, C, U, that's threonine. And then we get to our U, A, A. And in this one, it doesn't actually tell us to stop, but tyrine usually means stop. So in the one that you have in your notes, it does tell you stop. And that's where you stop. All right. Now I want you to try it on your own. Again, if you only ever do this with me giving you the answers, it's not going to be very helpful to you. You are going to have to do this in class for the next couple days and also on your quizzes and on your tests. So let's do this. So first off, we transcribed. So take a moment, make sure that you got the right answer. Pause the video. Now I want you to try to see if you can code out the amino acid sequences. Okay, let's see how you did. All right, if you got these, that means that you're on the right track. All right, now you'll notice that this one I was just trying to give you some more different amino acid sequences or amino acids to again sequence out. You'll notice that we didn't start with our AUG. Remember that that's usually how we did, and we didn't have a stop. Okay, that's totally fine. But again, normally when this happens, you always start with that AUG, and you'll always have a stop codon at some point. Okay. Now, let's end our very last page. Let's go on and wrap all of this up because I know that this was a really, really tough unit. So first off, let's talk about our central big concept of this unit, central dogma again. We started out this video with it. We're going to end with it. What is it and what does it mean? So the central dogma of biology is pretty much a set of known principles that make up the basis of biology. In other words, all biology is based on this because remember that all living things use the template that is DNA. And we also know that central dogma is directly correlated to DNA gives us RNA, which gives us protein. Now, you are going to need to know this and understand this concept very well, which is why you have an image on yours that looks like, again, this image that's right beside my video picture. And you'll notice that that's sort of complicated. You can't like draw that out. So you have a box in your packet. I want you to draw what I drew. That is doable. And you'll notice that there are a couple really good facts in there. First off, you take DNA. When you take DNA and you make more DNA, that's replication. When you take DNA and you make RNA, we call that transcription. When you take RNA and you make protein, that's called translation. You'll see that the DNA and the replication and transcription occurs in the nucleus, and that the translation to the protein occurs in the ribosome and the cytoplasm. And lastly, DNA never leaves the nucleus. This is the biggest point of why we actually have to even use RNA. If DNA could leave the nucleus, then, well, it could just make its own protein. But instead, it's stuck in the nucleus, and that's actually a good thing. The nucleus provides protection. It's less likely to get damaged. If it's constantly moving around and leaving, then we're going to have some problems. There's going to be some damage. The proteins aren't going to be made right, and the organism might not be able to be viable. All right? Now, this last part I want you to do on your own. This is pretty much summarizing central dogma. So take what you know, think about the processes, and try to fill in the blanks. I'm going to help you out with the very first one. DNA is replicated in the, where is it replicated? So I want you to finish this out. We will correct it next class, but see if you can do it on your own. If you can do it on your own, you've got a really great understanding of central dogma. So we're going to end here. Remember, if you have any questions, reach out to your teacher. Come to Biomaster Mondays, and have a great day. Bye.